During mitosis, replicated chromosomes are separated into two new nuclei. Before a cell divides and goes through mitosis and cytokinesis, it replicates the DNA during the S phase of interphase, starting with unduplicated chromosomes and ending with duplicated chromosomes. Use beads and assemble two sets of homologous, duplicated chromosomes with one red and one yellow partner. The connecting rod is the centromere. We start at the end of the interphase with 4 chromosomes. This cell has only 2 pairs of chromosomes compared to a human cell with 23 pairs. Chromosome pair 1. Place the centromere, plastic-coated magnet, in the middle of the chromosome. Use 6 red beads on one side of the centromeres and 7 red beads on the other side for each sister chromatid of the first member of the pair, and do the same in yellow beads for the sister chromatids of the second member of the pair of homologous chromosomes. Chromosome pair 2. Similarly, build a shorter pair with 3 and 4 red beads on each side of the centromere and one similar yellow chromosome.
Okay, so these here are 2 sets of chromosomes, and they are homolog chromosomes. So this one here is similar to that one. They're not identical, so they come from 2 different parents. And those 2 here are also homolog, so they're not identical. However, here, those sister chromatids, so this one here and this one here, they are identical. And same for those. Okay, now when you go through mitosis is just starting with 1 diploid cell and ending up with 2 diploid cells that are identical. So how are we going to do that?
Well, the sister chromatids are going to separate.
Okay, let's start with drawing a cell. This here is the nucleus, and then here we have the whole cell. What comes before mitosis?
Interphase.
So we are in late interphase, and now prophase starts. What happens during that time?
First thing, or one of the things, is the nucleus starts to break down.
Yeah, so
nuclear.
Nuclear membrane starts breaking down, and the chromosomes condense. Now we take off the whole nuclear membrane, and we just see the remnants thereof. And now chromosomes are condensed. What else do we see? Let's assume that this is an animal cell.
The spindle fibers start to form.
Okay, we'll take 2 spindle fibers. They usually form on opposite poles. So we have here the 2 opposite poles. And what's the next step? Prophase. Chromosomes are condensed, spindle fibers form. Now, what comes after prophase?
Metaphase.
Metaphase.
And line up in the middle of the cell.
They line up in the middle of the cell. So let's line them up here in the midline of the cell. So during the prophase, they don't have to necessarily be connected. So this is now metaphase. All the chromosomes are lined up in the middle of the cell. What helps them to line up in the middle of the cell?
Spindle fibers.
The spindle fibers. So we have spindle fibers kind of putting them towards the middle of the cell. There are spindle fibers that actually connect to each other. So, and then some of them connect to what part of the chromosomes?
The centromere.
So they connect to the centromeres, and those spindle fibers they actually make sure that the whole cell kind of keeps its shape. We have spindle fibers that are connecting to the chromosomes, and other spindle fibers that just connect to each other to give the cell the shape. And now the spindle fibers start pulling the chromosomes apart. The spindle fibers here shorten. And what is this phase now?
Anaphase.
Mm-hmm.
Yeah, so we are now here in anaphase. Chromosomes are being pulled apart. Then after anaphase, what's the next one?
Telophase.
Telophase. So in telophase, what happens there?
It starts to contract a ring a little bit, and both of the parts are at the poles.
Okay, so we pull the chromosomes towards the poles. Is this cell haploid or diploid at that point? It's a trick question.
Diploid.
It's still diploid. This here is a full chromosome, or all those 4 here are full chromosomes. Mm-hmm. However, they are now not duplicated chromosomes. They started as duplicated chromosomes, now they are unduplicated chromosomes. If we would put them together with the other part, we would call each of this a sister chromatide, but now they are unduplicated chromosomes. So now we put them back here, the spindle fibers vanish, then, as you said, the contractile ring forms. So this is a ring that kind of pinches the cell into two parts. Now there's another step that happens here in this region. First of all, the spindle fibers, they disintegrate. Then we get a new nuclear membrane, and the same then over here. Then the cell completely is being split apart, and then we have two diploid cells that are identical. Now the next step would be what? So we went to interphase, prophase, metaphase, anaphase, telophase, cytokinesis. So the step where the cell actually separates, it's cytokinesis. And then the next step would be, again,
Back to interphase.
back to interphase. Now, doing interphase, now those chromosomes start to duplicate again. So we have the DNA duplication. So we have those 4 chromosomes in the nucleus, and they now start to duplicate again. Okay, thank you so much.